Greetings, students. This is a biology revision class. This revision class is for grade 12. We are going to revise the biological molecules. The science of biology. Here, 4 sub-units are being covered. The first one is the scientific method. Second, the biological tools. Third, prevalence and promise of biological science. And the last one is HIV/AIDS. So before we consider the first, the scientific method, let's have some points to define some terminologies like biology. How is biology is defined? Biology is the science of life and living organisms. Here, living organisms are plants, animals, and microbes. Life is, it is simply, we cannot define it in a phrase or in a statement. Simply we are mentioning or the characteristics or the features of living organisms, that they have ability to reproduce, they can sense their environment, that is irritability, they can reproduce, they can excrete, they can grow, and the likes. So all these features goes to life. So biology is the study of life and living organisms. There are a number of fields of studies in the biology. To mention some of them, like zoology, botany, ecology, and other fields. There are a lot of fields of study in biology, like virology, herpetology, entomology. So all these are dealing with plants and animals study. There are others, a lot are remain behind. The other thing is, in this revision session, we are going to see again what science is. So science is a unique experimental system of acquiring a scientific knowledge. So to carry out the scientific process, scientists are going to follow different steps to arrive at scientific facts. These are asking questions first. Any biologist or any scientist explore about the natural phenomena, wants or she wants to ask question about the natural phenomena. For instance, how do plants photosynthesize? And how water is obtained from hydrogen and oxygen. So they carry out scientific investigations. How do lightning is occurring in nature? So and so forth. Different questions are coming in natural phenomena. So scientists, to answer to these natural phenomena questions, you try to follow the scientific procedures. The first one is asking questions. And second one is do background research. So what has been said about the scientific investigations by other scholars, other scientists. So many background research has to be carried out in a written form or any literature in the environment, any information in the environment, whether it is in a written or in oral or in different ways. So background research is the second step. The third one is construct hypothesis. This is what the scientists want to investigate. His statements are kept in a certain phrases. So the construction of hypothesis is coming at the third place. Then fourth, test with an experiment. So the hypothesis should be tested with experimental work in the lab. And next, there is analysis of results. The result that is obtained from the experimental work should be analyzed into certain data form. Then the investigator or the scientist arrive at conclusion. Then at the end of the conclusion, if the hypothesis is true, it is reported to the rest of the world through journals, scientific magazines, and the world is getting the information in a knowledge form. If the hypothesis is not true, the experimenter is try to construct another new hypothesis. Try again, whether it is partially true or completely false. So the researcher is constructing the hypothesis again and follow the whole steps again. So this way the scientists are carrying out the scientific method. Here there are 2 scientists to be mentioned are Redi, Francisco Redi and Louis Pasteur. These are the scientists who carried out a controlled experiment in a scientific way so as to disprove the theory of spontaneous generation. Earlier, before science is well developed, there is a theory, what we call it spontaneous generation. This theory says all about living organisms are coming from non-living organisms. So these theories and these ideas were disproved by the 2 experimental scientists, Francisco Redi and Louis Pasteur. So in their experiment, they disproved that living organisms are coming from, not from non-living organisms, it is from the living creature, not from the dead or the non-living creature. So they have carried out many experiments, and they have shown. Francisco Redi carried out on a piece of meat, the flies larvae are emerging. And the theory of spontaneous advocates, they say that if you keep it rotten meat for a longer period of time, then you could have flies from them. And this is disproved by Francisco Redi. Louis Pasteur also, there are some creatures in the air that spoil the wine. He carried out this experiment on the wine industry, that something is wrong with the wine and it tastes flat because of something which is not discovered yet. But Louis is the scientist that discovered there are microorganisms in the yeast in the wine industry that spoils the wine. So that this have been disproved for once and for all in the scientific world. So these are prominent scientists that disproves the theory of spontaneous generation. Louis Pasteur, to carry out his work, he used S-shaped flask, and that allowed to inlet the air. In the other one, he has made a curved flask. So in both cases, air can enter, but in the curved flask, there are microorganisms trapped by the moisture that is on the curved region of the neck of the flask. So the brose is not spoiled by the microorganisms. So this way they disproved, Francisco Redi and Louis Pasteur independently worked to disprove the theory of spontaneous generation. In doing so, in experimental work, there is always, when a scientist carry out an experiment, there are variables are there. If changes in the independent variable are shown to cause changes in the dependent variable, then the experiment can establish cause and effect. So always there is cause and effect in the scientific work. For instance, let's consider bacteria growth may be affected by moisture levels in the air. So here we have cause and effect. The cause is the bacteria, and the effect is that is the growth of bacteria that is affected by the moisture level. So the moisture level is the effect in this experimental work. The independent variable is the bacteria, the moisture level is the effect. So there are other variables also in line with this is the control variables. Variables other than moisture have been under control to make the result more fair and acceptable result. And the other one is the confounding variable in this experiment. That is, bacteria growth may be affected by the moisture level in the air. So the independent variable, as I've said earlier, the bacterial growth. The dependent variable is the moisture level, and the control variable in this experiment is all other features other than moisture will be under control. The confounding variable is the variable that is confusing with the existing variables or the independent variable. The independent variable is the bacterial growth. Moisture is an important for bacterial growth, but there are other factors might be mixed with this factor that is not clear for the experimenter. This variable, we call it the confounding variable, that is a confusing variable. Another one in experimental work, in a scientific experimental work, there are also accuracy, reliability, and validity. So what do we mean by accuracy in a scientific work, reliability, and validity? Let's consider them. Here, accuracy refers to how precisely a measurement is made. Example, we can take the graduated cylinder and non-graduated cylinder. So which one is accurately and precisely measure a certain volume of a liquid? Is a graduated cylinder or the non-graduated cylinder? Obviously, we have a graduated cylinder can give us, let's say, 10 milliliter of water. We can measure with a graduated cylinder. But in non-graduated cylinder, we don't know the volume of the exact volume of the water in this measuring apparatus. So in experimental work, precision and precise measurement is very crucial. Another thing in experimental work is reliability. This concerns how repeatable the results of an experiment are made and how dependable they are. So always the results should be consistent and it should be repeatable. For instance, let's consider starch test in carbohydrate. So to test this, we need iodine solution and a starch compounds, a starch molecule. We can take some foodstuffs like potato and bread, and we have a slice cut of potato, and if we drop iodine solution on it, we have a blue-black color. This is a test for carbohydrate existence. Starch presence in the potato or in the starch foodstuff. The same way, if you have a bread, moistened bread with water, and if you drop iodine solution, the same way you have blue-black color because the starch is there. So this is what we refer to reliability. Repeatedly, if you test this foodstuffs, you don't have the same blue-black color. So this is what we call it reliability. Validity is another variable that appears or that goes with the experimental science. Validity concerns whether an experiment really measures what it says it is measuring. Okay, so here we have controlled every aspects. We have said that moisture and other factors that are important for the growth of bacteria are under control, so that the result is become valid always when we measure the growth of bacteria. So validity is refers to experimentally what it measures always. It wants to measure. So the scientist is giving validity for his experiment always when he gets a consistent results by controlling other factors. So these are also appearing, this accuracy, reliability, and validity are also appearing in experimental work. Likewise, the cause and effect. Another important thing in the scientific work is the scientists get some results from their experimental work. They have to report to the scientific world. They are using different journals. For instance, journals like Nature and Science are mostly used for reporting the results of the scientific findings. So here the scientist is addressing the knowledge to the rest of the world. So the scientists in the world, in the same procedure, they try to repeat the experiment, and if they do have any doubt, they can ask the experimenter back again and to make improvement and accept some comments. So this way the works of the scientists are reaching for the scientific world. So these are an aspect of the scientific method. Another thing in the scientific method is any scientific report must contain scientific procedural works. So these procedures starts with writing a title page there, and then writing the hypothesis, a clear description of the experimental procedure, a full account of all the results obtained, conclusions, an evaluation of the procedure, and an acknowledgement. All these are the procedures in the scientific report. So any scientific report which misses one of or either of these will not be accepted. So in a scientific work, in a scientific method, all these procedures are very important. So with this, we end up the first session of the scientific method. Now we will consider the second aspect, that is the tools of biologists. So what are the tools of the biologist? Before we look at the tools of the biologist, and the biologist tools are classified into two. That is the one which are working in the field, and the other which are very important in the experimental lab. So both are different aspects, different ways the biologists use their apparatus or tools. To mention some of them, inside the laboratory, the biologists use instruments or apparatus like microscope, centrifuges, petri dishes, measuring cylinders, balances, and the likes are being used in the lab. So all these instruments do have their own specific tasks. Microscope used to magnify small creatures in different magnification size of the microscope. Centrifuge, this is to separate the solid and liquid separately. Petri dish, these are a biological tool where microorganisms can be grown with a certain kind of medium. A measuring cylinder to measure different volumes of liquid. Balances, this is to weigh some experimental substances in a liquid or a solid form. So they know the weight of the substance that they use in experimental work. Another one is the field or apparatus or field tools. These are like quadrants. These are a small metal frame, 1 meter by 1 meter or 2 meter by 2 meter, used to apply to know the size of the population of plant in a certain areas, let's say in a hectare of land. Plant press, this is important. The biologists go to the field with his plant press, cut some branches of plants and put them inside the plant press, keep it for a certain period of time until it dry out, and then label the name of the plant, the place where it found, who the person found these plants, the number of seeds, and the likes, the date press carried out, and the likes have been labeled, and this is kept for scientific further investigations and different scientific studies. Theodolites, these are biologists used in the field to measure the height of the areas. GPS, geographical positioning, biologists working in a water environment, in mountain areas, and lowland areas, and different areas, so who wants to know the position of the water. Flow meters, this is used in rivers, how fast the rivers flow. So the biologists used to measure the flow of water by the flow meters. So with this, we wind up the second session of the tools of the biologist. And now we proceed to the third part of the first session, that is relevance and promise of biological science. In this subunit, we consider a very important work of greater importance of biological science. Biological science is very important in agriculture, in medicine, in environment. Let's say step by step each of these categories, biology and agriculture. So the biologists are playing a great crucial role in addressing how to produce extra foods, let's say twice a year, So they select a good yielding crops, a disease resistant, a drought resistant. So for instance, if you consider Professor Gabbisa Ejeta made investigations on the sorghum bicolor, that drought resistant strain of sorghum is produced by these scientists and geneticists. So biologists are playing crucial role in this regard. The second one is biology and medicine. Also biologists are being trained as doctors and nurses, so play a greater role. And even they can give advice to government to reduce the rate of population growth, contraceptives, and educating people. So in this regard, biologists are playing crucial role in aspects of medicine. So doctors and nurses have been specialized for biological knowledge, and they can train with medical laboratory technicians, medical researchers, special researchers, for instance, research on cancer and drug development researchers. There are a number of biologists are working in a medical field. So biology is contributing to have a relevance for the environment, the people, and everything around in the living world. Biology is not determined with agriculture and medicine, but also it plays crucial role in the environment. Here, a biologist is playing in alleviating problems with global warming impacts. So this global warming is a long term of heating of the Earth's climate system. So how can this is alleviate? Or a biologist giving a solution for that. They will make advise governments, or they involve themselves in alleviating these problems. Very simply, planting trees will alleviate the global warmings. If you have many plants of plants around you, so carbon dioxide would be sunk into these plants and photosynthesis will carry out, so there's no problem. The global warming issue is the sunlight that reaches to the earth, and when it returns back, it is blocked by the greenhouse gas effects, the carbon dioxide, the nitrous oxide, the methane, and the likes. They block these gases and make heater the air. This will bring adverse environmental conditions. Some are heavy rain, some are drought, and some places in polar regions like the ice glaciers will melt down and the sea level rise. So different disturbances will occur in the world. So this is biologists are trying to give solution to global warming. Not only biologists are confined to global warming impacts, but also they care about species distribution in areas. They give emphasis what looks like, which plant species are highly dominating the area, which animals are rare, endangered. So biologists looking at different species, they try to make somewhat even, or make it a balance to the environment. Another aspect in the biology and the environment, they also conserve their environment by advising the governments of the local governments. So there are we are facing as an Ethiopian some floodings and erosions. Every year the land, the fertile land of the soil carried out to very far places. So from time to time the agricultural lands are degraded, they lose their minerals and the likes. So conservation is very important. Soil conservation, wildlife conservation, vegetation conservation, and air pollution conservations, how we can refresh our environment, prevent from polluting substances. So conservation is very important, and biologists are taking their leading role in this regard. And the other thing is introducing new species to the environment. For instance, we have African tulip tree. Now we have seen in very larger number around Addis. So this is introduced the species to the environment to balance the environment and to keep the environment with aesthetic values. So introducing new species of plants and animals for different biological purposes. So biologists are playing a great important thing in this regard. So it do have relevance with biological science. Another aspect of the biology is it has a promissory science. So the promise of research in biological field is very huge. We can see some of the promises that are carried out by the biological science. One of these is cures of genetic diseases. There are a number of genetic diseases are there. For instance, in human beings, the Down syndrome and the cystic fibrosis, the sickle cell, Tay-Sachs disease. So all these are genetic diseases are in the human population nowadays. So in the future, biologists are trying to alleviate the problems with regard to these genetic diseases. Another point is treatment for degenerative disease. There are a number of degenerative diseases are there, like the Parkinson disease, the Alzheimer's disease. The Parkinson disease are neurodegenerative disease, regardless with the neurons. The Alzheimer's diseases are destroy memory at all. So biologists are working seeking for treatment for degenerative disease. So this is promissory science in the future. We have the chance to get some remedies for these degenerative diseases. Biologists is giving promise biologically controlled industrial processes. There are industrial processes that carried out nowadays, like for instance, the industrial production of insulin. Using the hormone insulin is produced in a controlled manner in the biological way. Wastewater treatment, fermentation process are some of these biologically controlled industrial process. Biology has a vast thing to work in the future in this regard. The other one is tailor-made drugs. There is a promise with regard of biology and the future tailor-made drugs that directly fit to an individual for a specific kind of diseases, with a specific drug, with a specific dose, as the right patient, at the right time. So biology has a greater promise. The other one is genetically modifying plants. Biology in the future with a lot of modification of plants. Plants that are good yielding in dry conditions, so genetically can be modified. Plants with their own insecticides should be produced. So that is very important. Nowadays, insects are playing devastating role on the production of grains, crop plants. So this would be in the future with the work of biology might get some remedies and solutions. Cloning of productive animals and plants. So cloning, producing the same DNA organisms in a multiple copies. So these are very important in the spheres of animals, in the spheres of plants. For instance, strawberry plants, potatoes, they can be produced by cloning in a larger quantities to the ever increasing population of the world. Some animals like cats, dogs, horse, mule, ox, rabbit, and rats can be cloned. This is very important in our production of plants and animals to get maximum yields in a coffee bean production, for instance, in our country, and other products. Production of monoclonal antibodies, let's say, that can be delivered drugs for cancer cells using stem cells. These are undifferentiated cells, gradually differentiated to a certain kind of cell. So using this cell, there can be solutions for damaged organs, repairing damaged organs. New organs can be produced with using these stem cells. It can be tissues or organs or organisms can be produced using the stem cells. So biology has a lot of promises. Another important thing is the relevance of biology in Ethiopia. With regard to our country, biologists, any layperson must have the biological literacy. They understand about preservation, development, properties of the abundant biological resources. So biology in this regard, it do have a relevance to Ethiopia, to our country. And biology also do have a relevance combining the wells of the indigenous knowledge with the modern knowledge. We have a number of indigenous knowledges. We have talla making process, we have bread making, injera making, we have different kinds of herbal medicines, different traditional knowledges are there. These are combined with a modern knowledge so that it will produce a number of biologists that are very important for this country and give a lot of benefits. Another relevance of biology in Ethiopia is control land degradations. We have mentioned earlier, land degradation is a common feature in Ethiopia. So nowadays, many planting of trees we have seen at this time. That is good step for controlling the land degradation. This land degradation is not the land alone is degraded, but also there are a lot of biodiversity loss in connected to this will appear. So when we control land degradation, planting trees, and we take care of our environment, then plants can grow. Many animals are getting to this around the trees, around the grasslands, so that we are improving our environment and diseases and other problems will not face this country thanks to the work of biologists. So to develop the biological and agricultural potentials in this country, biology is playing relevance and important to this country. The last point of this session is biology and HIV/AIDS. Obviously, repeatedly we have mentioned in literature and always in media, this disease is still a pandemic. However, the disease is declining from time to time when we consider in terms of the taking care and some medications and protections. Obviously, we know that the AIDS is a disease, HIV is the virus, and the transmission is different routes are there, unprotected sex and syringes that are unprotected needles and unscreened blood and the likes can be the major cause for transmission. Maybe there are intravenous drug users are there. They can also be the case for transmitting the HIV/AIDS. And how do we prevent this? There are prevention from multiple sexual partner, and drugs such as high acute antiretroviral therapies are given. Combined drugs are given to minimize the virus spreading and prevent from or avoiding intravenous drug. Male circumcision, all these can prevent the HIV/AIDS. Communities do have playing a greater role in this regard. Care for each other, treat patients, avoid discrimination and stigmatizations. Communities also playing a great role. Nowadays, if we look at the 2 years back data of Ethiopian HIV/AIDS, 690,000 people were living with HIV. HIV incidence per 1,000 uninfected population over 1 year, 0.24. HIV prevalence, that is percentage of people living with HIV from 15 to 49 years, was 1%. 23,000 people were newly infected with HIV. That is reduced from 29,000. And 11,000 people died from AIDS related with the illness. That is from 20,000, the death is reduced to 11,000. So there are a great improvement in this regard. So this is the first session of the Science of Biology. And now we go to the second session, that is the biological molecule. The biological molecules are very important, as we know that plants and animals, microbes, they composed of these biological molecules. So these biological molecules are categorized as organic molecules and inorganic molecules. Before we separately look at these inorganic and organic molecules, let's say a few things. That organic molecules always contain carbon and hydrogen. The most common elements in living tissue are hydrogen, oxygen, nitrogen, and phosphorus. Atoms, elements, compounds are most of the components of the biological molecules. The simplest particle of elements are the atoms. Atoms contain one kind of atom. Compounds made from molecules containing atoms joined together in a fixed ratio. So these are a few introductory part of the biological molecule. Now let's look at the biological molecule, the inorganic one, water. Water is the most valuable and abundant molecule on the surface of the earth. So we're going to look at the nature of the water, its importance. Most abundant substance on earth, three-quarters of the earth's surface is water. Each water molecule joins one with the other with the hydrogen bonding. It has several properties that are vital to living things. Transparency is one property. The sunlight penetrates the water body in the ocean or river or lakes up to certain depths, and there photosynthesis can take place. And this allows many creatures to live inside the water bodies. So water is transparent and a transparency nature. So this is very important for living creature. Another property is high surface tension. The water molecules, they join together by hydrogen bond, and many creatures can live on the surface. Insects walk over it without any obstacles, and living organisms can live below the surface. So water has high surface tension, and this is very important for living creature. Another important property of water is a high specific heat capacity. So water does not heat up or cool down quickly. It is not easily become hot, or it's not become easily too cold. If you go to the daytime to the lakeside or riverside till noon, the water is cold. Gradually it heats and gradually it cools. This is important for living creatures. Another important unique property of the water is ice is less denser than the liquid water. Why this is so? Because the bonding of the hydrogen bonding is expand when it solidifies. This makes it lighter than the liquid water. Cools top down. This is another property of the water. The water forms ice not from the bottom of the water. Ice is formed from the surface down to the water body. This is an important property and quality of water to survive many creatures inside in it. If water makes ices from the bottom of the water, then many creatures are stuck in the ice glaciers. They lose many important minerals and foods, and even they will deprived of oxygen. So many life will die out. But this is not so in the case of ice formation in the water body. Ice is forming from the top to down. It is a good solvent, another property of water. Allow reaction to occur and making it ideal as a transport medium. So many substances get dissolved inside in it. So it is, most of the time, it is called as a universal solvent. Another property, it is reactant in many reactions. Okay, for instance, in photosynthesis, the reactant is water. Carbon dioxide react with water. In many digestion process, water is a reactant. For hydrolysis, in a photosynthesis, in a hydrolysis reaction, water is a reactant. It has a low, but not too low, viscosity, again making it ideal in transport medium. So many substances are transported with the water or with the aid of the water to inside the cell of the organisms. So far, students, we have discussed the inorganic molecules. Now we proceed to the organic ones. One of these organic molecules is the carbohydrate molecule. Carbohydrate is composed of carbon, hydrogen, and oxygen in the ratio of hydrogen to oxygen atom 2:1. And carbohydrates are classified as monosaccharide. These are carbohydrates with the atom. They are arranged in a single ring-like structure. The formula for the monosaccharide is alpha glucose, and all other hexoses monosaccharide is C6H12O6. Glucose, galactose are some of the examples of the monosaccharides. The second one is the disaccharide. These are two monosaccharide units are combined together, joined by condensation to form disaccharide. Example, maltose is two alpha glucose molecule joined with a loss of a molecule of water. The formula for maltose is C12H22O11. The bond joining the two molecules of the alpha glucose is an alpha 1,4-glycosidic bond. This is when two glucose molecules join together. One is in the first carbon, the other is in the fourth carbon. And the alpha is referring to the hydroxyl found above the ring. So it is said alpha 1,4-glycosidic bond. And another carbohydrate group is a polysaccharide, formed when many monosaccharide molecules joined by condensation. Examples, starch contains two polymers of alpha glucose molecule and amylopectin. Starch and glycogen are storage compounds. Starch for plants, glycogen for animals. They are a storage compound. They are stored in the liver and the muscle. In the case of animals, starch is in plants. They have compact molecule that enable much glucose to be stored in a small place. They are insoluble, meaning that they have no osmotic effect within cell and do not move from cell. Another organic molecule is example for this starch, glycogen, cellulose we can mention. And another organic molecule is amino acids. Contain atoms like carbon, hydrogen, oxygen, and nitrogen. Amino acids can be joined by condensation reaction. There is hydrolysis reaction in there. The bond between two amino acids is a peptide bond. A large number of amino acids joined this way form polypeptide. Polyproteins are polymers of amino acids. They have several levels of structures: primary structure, secondary structure, tertiary structure, and quaternary structure. Let's look this step by step. Primary structure is the sequence of amino acid in polypeptide chain. Example, insulin. This is a hormone. This insulin is protein and it is a primary structure. The sequence of amino acids in a polypeptide chain will give you primary structure. Secondary structure determined by folding of the primary structure into either an alpha helix or beta pleated sheet. Examples of the secondary structure are alpha keratin. This is found in hair and wool. Beta keratin in a beta pleated sheet. Silk proteins, fibrous proteins. So these structures are held in shape by hydrogen bonding. We can mention secondary structures found in bone, cartilage, tendon, skin, blood vessels, intracellular space. The tertiary structure, this one is the third structure of the protein, is determined by further folding of secondary structure into either fibrous or globular shape. These structures held in place by further hydrogen bonds, disulfide bridges, and ionic bonds. Example for this, globular protein enzymes. Typical example, enzymes are tertiary structures. Quaternary structure is the fourth structure of the protein in which two or more polypeptide chains came together, each with a tertiary structure, are bonded together. A hemoglobin molecule consists of 4 polypeptide chains bonded together. So hemoglobin is a typical example for the quaternary structure of protein. Another important point in our biological molecule, the organic molecule, is a triglyceride molecule. A triglyceride molecule is an ester of 3 fatty acid molecules and a 1 glycerol molecule. So a 3 fatty acid molecule combined with 1 glycerol molecule, we have a triglyceride molecule. The ester bonds are formed by condensation. So the bond joining these 2 molecules are the ester bond. So triglyceride is an important organic molecule. The fatty acid molecule is another organic molecule, can be either saturated or monosaturated, polysaturated, the fatty acids. So the saturated fatty acids, they have a single bond between carbon bonds. In a monosaturated, in a carbon-carbon double bond. In one carbon-carbon, you have a double bond. Then it becomes monosaturated fatty acid. You have more than one double bond in a carbon-carbon double bond, then we call it polysaturated. Another molecule, organic molecule, is a phospholipid molecule. This consists of 2 fatty acid and a phosphate group bonded to a molecule of glycerol. So 3 molecules combined together. 2 fatty acid, a phospholipid group, and a molecule of glycerol. This will give you a phospholipid molecule. This phosphate group gives a molecule hydrophilic head, and the fatty acid give the molecule the hydrophobic tail. The water-loving, the water headings are formed because of the phospholipid, these 3 molecules, the fatty acid, glycerol, and the phosphate groups. So phospholipid bilayers are the basis of biological membrane. So the biological membrane is made from this structure. And another organic molecule is the nucleic acid that is made from nucleotides, RNA and DNA. These are made from 4 nucleotides. And DNA is a double strand, RNA is a single strand. DNA has 4 nucleotides, but this is thiamine is replaced by uracil in RNA. RNA also do have 4 nucleotides. And DNA molecule is a stable molecule, RNA is an unstable molecule. And the sugar is in DNA is deoxyribose sugar. The sugar in RNA is the ribose sugar. So these are small discrepancies between nucleic acids, and these are also very important. The DNA is the blueprint of the genetic information is binded in this structure. The RNA is involved in a protein synthesis. These are important organic molecules. And we do have tests for some of the organic molecules in our lab. And these are carbohydrates, proteins, and fats. So how these molecules have been tested experimentally, whether a carbohydrate contains carbohydrate stuff, protein, fat, also fatty structures. So here, reducing sugar is non-reducing sugars are a test for carbohydrate, and proteins and lipids do have their own test. Reducing sugars are reacting with the Benedict solutions when heated to give yellow, orange, red precipitate. If it is yellow, the reducing sugar amount is little. Orange, medium, sugar is present. Red, completely sufficient sugar is present. So that these are the confirmatory tests for reducing sugar. Non-reducing sugar, it must be changed into reducing sugar. So it should be boiled with hydrochloric acid, neutralized by adding sodium bicarbonate, and then finally it is exposed for the Benedict test. Then the same color do you have. The non-reducing sugar examples like the sucrose, the reducing sugar, glucose examples. So sucrose must be changed into glucose to be tested with the Benedict solution. That is, in both cases, this confirmatory test refers to that these compounds, reducing sugar and non-reducing sugars, contains the carbohydrate stuff. Another test for protein is different tests available for protein: burette, xanthoprotic, Millian's reagent test. So 3 tests are there. When we look at the first one, the burette test, it reacts with burette reagents. The protein stuff is taken, like the egg albumin, the white egg albumin. When it reacts with the burette reagent, it gives mauve or purple color. That is a confirmatory test. In a xanthoprotic test, when protein is existing in a certain foodstuff, yellow color is a confirmatory test. And if it is a Millian's reagent, then red color is a confirmatory test for protein. The fats and lipid test is an emulsion test, we call it. So here, the test for the lipids is, for instance, oil and fats. If you take oil, alcohol over it, that is methanol, then you vigorously shake it, then allow to settle down, then take the decant, add a drop of water sufficiently, then you do have a milky white color. This is a confirmatory test for fats. Sometimes you do have a greasy spot test. You drop an oil on a white paper remains transparent. That is also another test. The third test for the fatty substance is Sudan-3 test. This is a compound added on a oil and it gives red color. That is a confirmatory test for the carbohydrates. Students, in this revision class, we have raised the organic and inorganic molecules. I hope you have enjoyed the lesson. Till we see in the next lesson, goodbye.